All right, this next section of the lecture, 14.3, involves the systems that are part of the immune defenses. So we're going to have an overview of a lot of the tissues and organs in the cells that are part of the immune defense. That will be before the second line of defense that we're going to be discussing in section 14.4, I believe. So now, we're going to be looking at the compartments that are going to participate in immune function. We're going to be looking at mononuclear phagocyte systems, the system of cells that are going to be gobbling bacteria up, the spaces surrounding the tissue that contains the extracellular fluid, the bloodstream, and the lymphatic system, which are the two circulatory systems in the body. So now, let's look at the macroscopic level. What we have here in the right is a picture of the tissue. We have blood capillaries that are going to allow fluid to go in and out of the system with oxygen that is going to be given to the cells and removing carbon dioxide. We're going to have macrophage cells shown over here, for example, that are going to be scavenging the area around, looking for microorganisms and debris to eat. Reticular connective tissue, shown here as these lines, that are going to be connecting the cells and providing a matrix for the cells to hang out. We have here the dendritic cell, which is also like the macrophage, picking up particles that are present in the liquid that is here in the extracellular fluid, shown in here in this area as pink, and is able then to collect it to show to the immune system. Of course, here is a tissue cell of that particular tissue. It could be any of the tissues that we're talking about. Not very far from the capillary system, we have the lymphatic capillaries, which are going to be carrying lymph. And the lymph we're going to see that is going to be the second circulatory system. So the blood and the lymphatic capillaries are going to be present. They're going to be circulated about differently. The capillary system using the heart and the lymphatic system using the muscle contractions of the body. And what we're going to look are the cells and chemicals that originate in the reticular system. Those are going to diffuse in the extracellular fluid that is going to migrate into the blood to be carried by the heart into the spleen or be carried through the lymphatic capillaries to the lymph nodes. So the flow of events is going to depend on where an infectious agent starts first. And let's take a typical progression of that system. So we're going to have to break down the first line of defense, the skin or the epithelium. So we're going to have now that invader invading the extracellular spaces and the reticular tissues. You're going to have lymphatic circulation, so the cells in the lymphocytes either escaping the blood system or the lymphatics can come in, and eventually that leads to the bloodstream. So we're going to take a look at that in more detail. First, let's learn a little bit about the mononuclear phagocyte systems, which are going to include cells that are going to phagocytose. And this is a network of connective tissue fibers that are going to encase a lot of mononuclear cells. Mononuclear because when we look at them on the microscope, they seem to have one nucleus, as opposed to lymph cells that are called the polymorphonuclear cells that looks like their nucleus is divided into 3 or more pieces. Now, this system is going to have a basal lamina, which it's going to serve as attachment to the reticular system cells, and is interconnected by many different cells. And you have that massive mesh of connective tissue that is surrounding all the organs, the extracellular matrix. In there, we have macrophages that, as I showed you earlier, are going to be scavenging around in the system. And the macrophage systems are going to have a different name depending on the tissue that they're found. For example, macrophages found in the brain are called microglia. In the blood, before they exit, are called monocytes. Macrophages can live in the as activated macrophages in tissue. Coffer cells are going to be found in the liver. Histiocytes are going to be found in connective tissue. Glomerular mesenchymal cells, mesangial cells, glomerular mesangial cells are going to be found in the kidneys. Synovial macrophages are going to be found in the synovial fluid in the joints. Splenic macrophages are going to be found in the spleen. Pleural cavity macrophages are going to be found in the pleural cavity. Osteoclast inside bones, alveolar macrophages in the lungs, and lymph node macrophages in many of the lymph nodes. So depending on who discovered them, they may give them a different name, but all of them tend to be family of macrophages, and they have a precursor that is the blood monocyte. So when the monocytes are in the blood, they're called monocytes, and once they get out of the blood into a tissue, they will adopt a different name like macroglia or macrophages. So as part of the mononuclear phagocyte system, we can look at them exploring many different areas. And we talked about that they're going to be exploring the mucosa. So we have, for example, two examples of mucosa here. The gut-associated lymphoid tissue, or GALT, which is the lymphoid-associated tissue in the gut and digestive tract, and the mucosa-associated lymphoid tissue, which is the more overall system, which is now called the MALT, which is found in all the tissues that enclose mucosa. These provide a major function of testing these different tissues for foreign invaders and alerting the immune system about them. So they have an auxiliary route to return the extracellular fluid into the circulatory system, and it serves as a drain-off system for the inflammatory molecules. So something happening in an inflammatory tissue will then get dispersed systemically. So there is a surveillance system, so they can render surveillance and recognition and protection against foreign materials that are invading through any of the mucosa of the lung, the respiratory system, including the lung, the gut system, the skin system, and the urogenital tract. So they include lymphocytes, phagocytes, and antibodies produced by lymphocytes. So the lymph, it's going to be part of this lymphatic fluid. And it is a fluid that is carried by the lymphatic system, the second circulatory system in our body, composed of proteins and many different salts diluted in an aqueous solution. And it's able to serve as an information highway for white blood cells, for fat, for cellular debris, and that's very good because it transfers the cellular debris to the lymph nodes to be exposed to the immune system. And of course also the infection agents that are infecting the extremities. So as I mentioned to you in lecture, if you have a cut in the arm, the material from that cut in the arm will be transferred through the lymphatic system all the way to the armpit lymph nodes, where it can then be exposed to the immune system. So here's two differences between the blood and the lymphatic system. So the blood, it's moved by the heart. So it's going to be moving in one direction by the heart. The lymph, however, moves in one direction only, from the extremities to the heart. And it is pushed by the muscular system that it's allowing us to move. So as we're moving, walking, waving our arms, that is going to help to move the lymph. The blood system moves from the heart to the extremities and from the extremities back to the heart. So that is a very big difference. So here we have a picture of both the circulatory and the lymphatic system. As you're very well aware, the heart is the major pumping system of the circulatory system and is going to pump the blood from the heart into the lungs, where it's oxygenated, and from the lungs is going to be distributed to all the extremities. On the other hand, on the right, we have an image of all the lymphatic system, where the fluid via the lymph is going to be moved from the extremities to the lymph node and then inside the heart. So the arms will drain here to the armpit lymph nodes, the legs will drain here to the groin lymph nodes, and we'll also find lymph nodes in other areas like the GALT, the gut-associated lymphoid system, the spleen, as well as the mammary glands, and the ones in the throat, including the cervical nodes and the tonsils. So what can we tell you more about the lymphatics and circulatory system? So here again we have the issue of the comparison of the movement. So as I mentioned to you, the heart is going to be pumping blood that will go from the heart to the lungs and then be carried by the arteries all the way down to the extremities, and then in the capillaries is going to be exchanged to the veins, and the veins are able to bring that blood back to the heart where it will get reoxygenated in the lungs. The lymphatic system, on the other hand, is working by gathering the lymph from the extremities, passing them through the lymph nodes, and eventually having a joint portion where it goes back into the heart. So the comparison of the generalized systems, both of them are parallel to one another, but the transfer of the lymphatic system is in one direction, whereas the transfer of the blood goes from the heart to the extremities and back to the heart. So now, let's take a look at some of the tissues that are part of the lymphoid system. We're going to get a quick look at the thymus, which is the center for many of the white blood lymphocytes called T cells are born. We're going to take a look at the lymph nodes, where a lot of the filtering from the lymph is happening. We're going to take a look at the spleen, where the blood is filtered, and we're going to take a look at the MALT, the mucosa-associated lymphoid tissue, which is the general term for all the endothelial tissue exposed to the environment. Now, the thymus, it is oftentimes people call it an endocrine system, but it's really not. It's actually an immune system organ, and it's right on top of the heart. In babies, it's very large, and it's composed of two different tissue types. We have the cortex that is here shown in dark pink around the edges of the thymus, and the middle part of it has the medulla. It has blood vessels and capillary vessels that are going to be allowing the oxygenation and the removal of waste from the thymus. And in small children, it's very large. And as the child grows, it becomes smaller and smaller until it is actually very small and almost non-existent in adults. So the largest shore of growth, it happens in puberty, and eventually in adults it grows down and shrinks. But it's the place where the adaptive immune system composed of T cells is going to be generated. So it's one of the major organs of the immune system. The lymph nodes are a network of small bean-shaped organs that are dispersed all over the body, where the extremities and the trunk meets. So as you can see, we have a lot of lymph nodes in the neck area and the collarbone. We have lymph nodes in the armpits that are going to drain the lymph from the arms. And we have lymph nodes in the groin area that are going to drain the lymph from the legs. And they tend to be the area where the molecules and particles that the lymph gathers are able to get filtrated and shown to the immune system. So it's a very large network of filterable units that are going to gather infectious material and be able to show it to the immune system. And whenever there is inflammation, the lymph nodes tend to enlarge as the cells of the immune system come in and populate it, recognizing the foreign invaders. The spleen, on the other hand, is another filtering mechanism, but instead of filtering the lymph, it's filtering blood. And its primary function is to remove dead cells that are going to get out of circulation. So if there's a lot of red blood cells that are dying, they get removed by the spleen. However, it also has an immunological function, like the lymph nodes, to be able to filter pathogens that are found in the blood and show it to immune cells so they can launch an immune response. So it is the blood equivalent of the lymph nodes. Whereas the lymph nodes are going to filter the lymph, the spleen is going to filter the blood. Now, we have a lot of smaller lymphoid tissue that is found in the tonsils, in the breast, and in the MALT-associated tissues. So the GOLD, that's the gut-associated lymphoid tissue. The MALT is the mucosa. That's the general term for all the other ones. SALT is the lymphoid tissue associated with the skin. And the BALT is the immune-associated tissue of the bronchia and the lungs. In the intestines, we have hundreds of Peyer's patches, which are small lymph node-like structures that are picking up material from the intestine to show to the immune system in case that there's infection on the gut. And it serves to also control and recognize the particles that could be form allergic reactions in foods. Now, together, all of those are going to be the tissues that control the immune response. Now, let's go back to the blood and check out the two components, because the blood is the main fluid that is circulating in the circulatory system and is composed of three major components. You have the plasma, which is the liquid component of the blood. You have the red blood cells, which are the major cellular components of the red blood cells. And then when we separate it by centrifugation, if we separate it by centrifugation, we divide the blood into plasma, red blood cells, and a small layer of cells called the buffy coat. And that buffy coat is where you have the white blood cells. So oftentimes when we do research in blood, we separate the blood, remove the plasma, get the buffy coat, and eliminate the red blood cells that we want to do it. Of course, you can clot the plasma and now obtain the serum. The plasma and the serum, it's the same thing except that the clotting factors have been taken out. So the serum then can be used to be transferred, for example, to another person without having the clotting factors present. And we use that oftentimes, both plasma and serum, we can use it to do immune testings, to check if there's antibodies against a particular infection, or if there has been a kind of therapy and we can measure it, the levels of it in serum and plasma. So when we think of plasma, it's mostly water, but it has a lot of the important proteins that are involved in the respiration and carrier of oxygen. So we have the albumins and the globulins that are carrying blood, but you also have the major antimicrobial proteins called antibodies. It also has a lot of the clotting factors and molecules like fibrinogen that are able to opsonize, which means cover a microorganism in proteins to target it to the immune system. Of course, it is the main the plasma, it's the main conduit for the movement of hormones from the endocrine system. And because it is the same kind of system that is carrying oxygen and nutrients to cells, including glucose or amino acid or fatty acids and ions, is also carrying the waste products and the CO2 that the lungs and the kidneys are able to filter out. So it is an important component, and as we mentioned in class, it is one that could also be infected by microorganisms. So let's take a look at some of the white blood cells that are found in that buffy coat that I mentioned to you. And we call it, in general, leukocytes. So they are different because when we separate the blood, as I showed you in the previous image, they lay as a small layer of white cells on the top of the red blood cell band in a separated blood sample. And they're going to include the neutrophils-like cells shown over here. Remember that I told you that they're called polymorphonuclear cells because look at the nucleus inside of this cell. It looks like it has 3 nuclei instead of 1. So it's one nucleus divided into 3 portions. Eosinophil, another member of the same family, also have the polymorphonuclear shape to the nucleus. What you can appreciate from here are this dot inside, which are full of granules. And another cell that is also polymorphonuclear is the basophil, but this one has many more granules inside. That is in opposite to the monocytes and the lymphocytes that are monomorphonuclear because you only have one shape of a nucleus shown over here and this one over there. The monocytes tend to be very large with a very large cytoplasm. The lymphocytes tend to be smaller with a very small cytoplasm. And not very far, you have a platelet right there. That little guy there is the platelet, which is a cell involved in the clotting of cells. So when we look at this, we can have granulocytes, which have a lot of granules. That is going to include the eosinophil, neutrophil, and basophil, and the agranulocyte cells, which are going to be the macrophages and many of the lymphocytes. Not all of them, but many of the lymphocytes. So again, looking at this, guys, we have the granulocytes, the neutrophils, eosinophils, and basophils. When you test them with eosin, they stain beautiful in blue and pink. And you can see the granules inside of the cells, here in pink and here in blue. So those granules contain a lot of the molecules used to kill bacteria. And in here on the right, you have a little bit of the dynamics and composition on the blood. Neutrophils tend to be a very large composition of the circulating blood, especially in patients with large infections. They tend to have a lot of neutrophils. And in those granules, they have the toxic chemicals that led them to kill bacteria. But these cells also are very good at phagocyzing bacteria and other parasites. Eosinophils also have granules inside, but those granules contain other molecules like peroxidases, the enzyme lysozyme, and some other digestive enzymes. They have toxic proteins and inflammatory chemicals that are able to elicit an inflammatory response. And they're really good at destroying large eukaryotic pathogens like worms. So it's one of the things that they're good about doing. Basophils tend to be a minority of the blood, about 0.5 of the white blood cells circulating. And one particular group of basophils called mast cells are very important in destroying also worm infections. So when we think of the other type of cells, the agranulocytes, because they don't have any granules, those tend to be the lymphocytes and the monocytes. But again, do not think of lymphocytes as being completely agranulate lymphocytes. Some of them do contain cytoplasmic granules. We're going to be looking at the natural killer cells, for example. They do have granules, especially when they're activated, and the activated CD8 T cells. But when we look at them in general, now we have between 20 to 35% of the circulating white blood cells. And 1 in 10 of all the adult body cells tend to be lymphocytes. They are the main fighters of the immune system. And they come in 3 different types. The B cells that produce antibodies, the T cells that are able to recognize viral infected cells and tumors, and the natural killer cells, which tend to work more with the innate immune system, which are also good at recognizing viral infected cells and tumors. T cells and B cells are going to be more part of the adaptive immune system, which we're going to learn a bit later in chapter 15. So in a nutshell, just to show you a little bit about them, B cells are the cells that produce antibodies. As I've shown here in this picture, the B cell has antibodies on its surface. When it binds, for example, to a bacteria, this bacteria can trigger the B cell now to become what is called a plasma cell. And the plasma cell's all purpose is life. It's going to be to secrete antibodies that are then able to specifically target the bacteria that it recognizes. So this is part of the humoral response or antibody-mediated response, and it's extremely specific. On the other hand, T cells are also very specific, but they tend to be doing hand-to-hand combat. Whereas you can think of antibodies as being a missile that can be launched for a distance and eventually it finds its target, the T cells tend to go and interact directly with the cell that they want to destroy. So for example, in this picture here is a cytotoxic T cell with some granules inside. As I mentioned to you, some of them do have granules. And they're able to recognize the infected cell by looking at some foreign particles made by the infected agent inside the cell. Once that recognition happens, the cell is able to release the granules that it has, and those granules are able then to release their toxic components to destroy the infecting cell. And therefore now that cytotoxic T cell, it's ready to go and attack other cells that are infected by the same pathogen. But again, they tend to be very specific. A T cell that is attacking a cell infected, for example, by the influenza virus is not going to be able to attack or destroy a cell infected by the Zika virus, because there's only cells trained to recognize influenza will then be able to attack influenza infected cells, but not Zika infected cells. So this is part of that specific immune response that I tell you that we're going to check in chapter 15. So let's continue now with some of the non-specific lymphocytes, which are going to be the monocytes and the macrophages. And I gave you a little bit of an introduction about the monocytes. They are the macrophages that are moving in blood. They are created in the bone marrow, and they move from the bone marrow into the bloodstream, where they will circulate and eventually exit into a tissue. So once they are left the circulation, they become macrophages, and the macrophages are going to have a different name depending on the tissue that they are now going to live in and stay there for a very long time, scavenging and clearing particles, dead cells, as well as infection agents that are going to infect that tissue. So what do? They have some specific functions, but they also have a lot of non-specific functions. They help with cellular housekeeping. They're going around cleaning debris and damage that are created by the inflammation and the infection. They're really good at processing some of those foreign molecules that are being shown by bacteria and viruses and then showing them to the immune system to activate it. And they're able to secrete biologically active compounds, which you're going to call them, for example, fever and inflammation cytokines, so the hormones of the immune system, that they can assist, mediate, attract, and inhibit immune cells and reactions. So this little picture at the bottom shows you some of those functions. They can kill microbes, they can kill tumor cells, they can activate neutrophils via some of these activating molecules like interleukin-8 or tumor necrosis factor alpha. They can help in tissue repair and organization. A side effect of that tissue repair and organization can be tissue damage, especially if they're causing inflammation and fever. And they're able to also activate lymphocytes. So they have a lot of different specific and non-specific functions. Now, a specific kind of cell, it's the dendritic cell shown here in the right. It's called a dendritic cell because it contains dendrites, which are these long elongations of the membranes, which in reality what they do is that they increase the size of the surface area of the membrane. And they tend to be able to live in tissues, but once they find a molecule that activates them, they are moved then into the lymphatics, either into the spleen or the lymph node, where they can then activate the immune system. So they can ingest bacteria and viruses that are going to stimulate them, and then once they are activated, they move to the spleen or the lymph node, and then they show the specific cells of the immune system, the B cells or T cells, like, hey, I found this bacteria, I found this virus in the leg, there's something going on there, go keep a look at it and check it out. So that is the major function of the dendritic cell. It tends to be a scavenger and a snitch, and it snitches on the bacterial and viral infection. It's like the Paul Revere of the immune system, where it's going to say, you know what, I found this, there's an attack, come and help fight it. Other cells that are found in blood are going to be the erythrocytes, the red blood cells, that are going to help carry oxygen into the tissues and carbon dioxide out. They do not have an immune system function per se, but they're designed to help with the oxygenation of the tissues. And the other ones are the platelets, which are fragments of cells from something called a megakaryocyte, and they help in forming blood clots and sealing areas that have been damaged. So they're very useful, and we often look at platelet counts to see how healthy our immune system and blood system is. So low platelet count means that there's something going on with the bone marrow that is not making sufficient of the blood cells, and we can use the platelet counts as part of that. And with that, I'm going to finish this part and this video.